Hi, welcome to biology classes with Lakshmi Chandra. We had completed the whole aspects of DNA as our genetic material. But then how it was found out that DNA is the genetic material? Let's focus on that today. Our topic is the search for genetic material. So how it was found out that DNA is genetic material? There were many experiments behind. We learned 3 very important experiments which led to the fact that DNA is the genetic material. Let's go back a little bit to our Mendelian genetics. Mendel found out that there are some factors which are passed on from parents to progeny, and those factors are actually our genetic material. But he didn't know anything about DNA. Later, Thomas Hunt Morgan and his team, they found out the chromosomal basis of inheritance. That means they found out that genetic material or the factors which Mendel had told is actually there present in the chromosomes. So the attention or the focus of the scientific community who were actually in a quest or in a search for this genetic material was all with respect to the chromosomes, or they were going down to the molecules to find out which is our genetic material. So in this chapter, this molecular basis of inheritance, we are going to deal with the topic how it was found out it is the molecule of DNA that is the genetic material. Okay, so 3 very important experiments we'll focus on. Let's first see the experiments done by a scientist or a British medical officer rather named Fredrick Griffith. Fredrick Griffith in the year 1928 is actually a British medical officer. Okay, a doctor. So he was doing a small experiment on 2 different strains of a bacterium. And the name of the bacterium is streptococcus pneumoniae. From this word, the name of the bacterium, streptococcus pneumoniae. Always whenever you write scientific names, you have to start the genus name with a capital letter, species with this name with a small letter, and you have to underline the genus name and the species name separately. So this is part of a binomial nomenclature. You must all know this. You must all keep this in mind, because children, if these questions are asked for 1 mark, name the experimental organism of Fredrick Griffith. If the question is like that, and if you have written the answer very well, it is streptococcus pneumoniae, spelling everything is correct, but then you didn't actually observe this scientific nomenclature, this binomial nomenclature, that particular things which you have learned in standard 9, if you haven't referred to that here in your answer, or if that is not reflected here in your answer, you will definitely lose marks. So I don't want any of my students to lose marks on such small things. So you have to keep this in mind, children, whenever you write a scientific name. In this NCERT textbook +2, there are so many areas where you come across the scientific names. So whenever that is asked, keep this in mind. Genus name should begin with a capital letter, species name with a small letter, and they both have to be underlined separately. Okay, so Fredrick Griffith was working on streptococcus pneumoniae, a bacterium. This bacterium causes pneumonia, as the name suggests. Okay, and see the streptococcus, that means it's a strip of round cells. Coccus means round in shape, isn't it? So strip means one after the other. The round cells are connected down. So its nature is like that. So Fredrick Griffith was working on streptococcus pneumoniae. And he observed that there are 2 strains. two strains or two types or two forms of this particular bacterium, streptococcus pneumoniae. One had a smooth appearance. One had a very smooth appearance. The other one had a rough surface. The other one had a rough appearance. Two strains, smooth and rough strain. He called this as the S strain, and this one he called as the R strain. Two different strains. Do you know why this is smooth and this is rough? Actually, the smooth strain is also having this rough structure, but around this rough structure it is having a coating of a polysaccharide. It is having a polysaccharide coat. A polysaccharide coat, or it is having a polysaccharide mucus in nature. Okay, slimy, slippery. So it is having a coat over this one. So that is why it is appearing as smooth. But this one is not having that coat. So it appears rough. So Fredrick Griffith was experimenting on this particular bacterium, Streptococcus pneumoniae, and he found that there are two strains. One is having a smooth appearance, he called as a S strain, and the other one, it is not having that coat, so that had a rough appearance, so he called as the R strain. Now he wanted to find out which among these two strains is actually responsible for causing the disease pneumonia. So what he did, he started doing experiments with the mice. Let's focus on that. His experiments. Okay, experiments of Fredrick Griffith. So his question is, which one is causing the disease? So what he did is, first set of experiments, he took S strain and he injected this S strain into a mice. He injected this into the mice. And he found that the mice got the disease pneumonia, and because of that, ultimately the mice was dead. Again, he did the same thing with the R strain. That means with the rough strain. He injected the rough strain also into a mice, into a healthy mice. But then this R strain, when it was injected into the mice did not get the disease pneumonia, and this mice, it survived. It survived. Okay, so from this, what did he conclude? He concluded that it is the S strain which is causing the disease, or he concluded that S strain is the virulent form. Virulent means disease causing form. It is the S strain which is actually causing the disease. So R strain is not causing any disease. So this is the first step of experiments that he did. And the second step, what he did is, he took heat killed S strain. What is this? Heat killed S strain. He took a few colonies of S strain bacteria, okay, and he subjected that to heat treatment. High temperature followed by low temperature, then again high and then low. So intermittent temperature he kept on treating. So heat shocks he used to give. Okay, so that is called as, and because of that, the whole bacteria, they got dead. So that is called as heat killed. So how did he kill it? By giving heat treatment or heat shocks. Okay, so the S strain was heat killed, and this heat killed S strain was again same thing, injected into a healthy mice. The mice survived. That means if the S strains are heat killed, it is not able to cause the disease. Now same thing he did with R strain also. Heat killed R strain. Again was injected into another healthy mice. Naturally, a live R cell is not able to cause the disease. Same way, a heat killed R cell will never cause the disease, isn't it? So in that case also the mice survived. So from this he was very sure that only a live S strain, a living S strain, a normal living S strain can only cause this particular disease. The third set of experiments that he did was very interesting one. Let's focus on that. In this third set of experiments, what he did now, he mixed live R strain. R strain is alive. Live R strain. He mixed this with heat killed. I am writing here heat killed as HK. Okay. He mixed this with heat killed S strain. That is, live R strain is mixed with heat killed S strain. And he kept it like this for a few days. And after some time, he injected that particular combination into a mice. He mixed these two. Which two? The R strain, the live R strain, and heat killed S strain. So what he was expecting in both cases, that is R strain, if it is injected, the mice survived. Same way, if it is heat killed S strain, again the mice survived. So when he had injected these two, naturally the mice should survive, isn't it? But then the mice was dead. The mice was dead because of pneumonia. The mice got the disease pneumonia and it was dead. And he was repeatedly doing this, thinking that he had made some mistake in the experiment. So very carefully he was doing all these steps again and again. And every time he got the same result. The mice was dead because of pneumonia. And he was curious enough to find out why the mice got the disease. Because in the previous two experiments, the mice survived. When this was injected separately, the mice survived. But then what is happening when this was mixed and it was injected? So he wanted to find out. And what he did is, he extracted the blood of this dead mice and he found out which bacteria is persisting there. And from the blood of this dead mice, he could isolate live S strain. See the miracle. Which one did he inject? He injected heat killed S strain. What did he get? He got live S strain. How can a dead organism become alive? It's not possible. But then again he repeatedly did this experiment. Every time he got the same result, the mice was dead. And every time he could isolate live S strain. So he was very sure that his experiments did not go wrong. But then something is there. Some molecule is there which is actually converting this live R strain into live S strain. Something is converting live R to live S. Never a killed organism can become alive. But then this live organism is getting converted to a live S. So live R is getting transformed to live S. He gave that word. Live R is getting transformed to live S because there is some transforming principle which is moving from live R to live S. Because why did he say so? What is the difference between these two? What is live R actually? Live R is having a rough surface. isn't it? How is this becoming smooth? Because it is having an additional polysaccharide coat. So something is moving from the heat-killed S strain, it is moving into the live R strain, and the live R strain is getting converted to live S. Something is moving from the heat-killed S strain. The heat-killed S strain is killed. You have applied heat and it is killed. But still, some molecule is surviving that heat shock. It is very stable, that is why it is able to survive. It is surviving that heat shock, or it is surpassing that heat shock, and it is able to enter into this live R when it is mixed and kept. Okay, and that molecule is converting the live R to live S, or instead of converting, he used the word transformed the live R to live S. And he told that is the transforming principle. Transforming principle. That is a transforming principle. So Fredrick Griffith put forward the transforming principle through these experiments of streptococcus pneumoniae and mice. Okay, what did he do? He first wanted to find out which is the virulent form. He found out that. Then he mixed the two non-virulent forms together, live R and heat-killed S. Then what did he get? He got live S from the mice blood which was dead. And what did he say? He told that some molecule is transforming the live R to live S. That molecule is coming from this heat-killed S and is converting this live R to live S, and that molecule is called as the transforming principle by Fredrick Griffith. Having seen the experiments of Fredrick Griffith, so in that experiment he found out that there is something which is getting transferred from heat-killed S cell to live R cell, and it is converting live R to live S, and he called that as transforming principle. It was in the year 1928. Later on, there were three scientists named Oswald Avery, Colin MacLeod, and Macklin McCarty. Avery, MacLeod and McCarty. So Avery, MacLeod and McCarty, they were trying to find out the biochemical nature of transforming principle, or biochemical characterization of transforming principle was done by them. They wanted to find out what is the transforming principle. Fredrick Griffith gave the word like transforming principle. Okay. These three scientists, they were behind that transforming principle. They wanted to find out which is the molecule that is actually converting this heat-killed S, which is transferred from heat-killed S strain going into the live R and converting the live R to live S. They wanted to find out. So what they did is, they isolated the different biomolecules. Like they isolated the proteins, they isolated RNA, and they also isolated DNA molecules from the heat-killed S strain. So all these molecules were isolated from heat-killed S strain. Just correct and write it. All these molecules were isolated one after the other from heat-killed S strain. Okay. And one by one, these molecules were injected into, or it was mixed with, the live R strain. It was injected into the live R strain one by one. So protein was injected and they were trying to find out whether this live R is getting converted to live S. So in the first case, when protein was injected, nothing happened to the live R. live R strain was remaining as R strain only. Next case, they injected RNA into the R strain. Was there any change? No. The R strain was remaining as R strain. Now finally, they injected the DNA molecule into this live R strain. But then the live R was converted to, yes, it was converted to the live S strain. So they concluded that it is the DNA which is actually converting this. It is the DNA which is converting this live R to live S. It is the DNA from the heat-killed S strain which is entering into this live R and converting this live R to live S. And they told that it is the DNA which is the transforming principle. So likewise they gave the biochemical characterization, or which is the biomolecule. They conclude that it is the DNA. But again they wanted to confirm their conclusion. They wanted to confirm it. For that what they did is, Avery McCarty and MacLeod, to confirm this particular molecule, that it is DNA which is transforming, to confirm that, they took proteins plus RNA plus DNA, all these three molecules were taken together, okay, and to all these three molecules they added also live R strain. So live R strain was there in a culture plate. Along with that, what did they add? They had taken the protein, the RNA and DNA all together was added into this. Okay, so like this, they took three different arrangements. In all these three, the arrangement is same. Protein is there, RNA is there, DNA is there, plus live R strain is there. The third case also protein, RNA, DNA plus live R strain. All three cases it was the same. But then what they did is, in the first case here, they added an enzyme. They added an enzyme called as protease. Protease. See the enzyme protease. Protease are the enzymes which digest proteins. They will cut these proteins into parts. They will cut it into pieces. They will digest the protein. So such enzymes are called as proteases. So when they add proteases, nothing happened to this. That means here live R was getting converted to live S. Why? DNA is here. DNA is intact. There is no change happening. Already they found out that DNA is the one which is doing this conversion. But they wanted to confirm it. Okay, to confirm it, they added one by one. They took these three conditions same and they added the enzyme protease. So all the proteins were digested. But still this conversion took place. Which conversion? R was converted to S. In the second case, the second experiment, they added the enzyme RNase. So RNase will digest all the RNA molecules. Okay, but still the live R was converted to S strain. This conversion happened. But in the third case, what they did is, they added the enzyme DNase. This DNase actually digest DNA molecule. They digest DNA molecule. And so, live R was not converted. It was not converted to S. Why live R was not converted to S? Because what is getting digested? DNA is getting digested. Since DNA, they found out DNA is the one which is entering, and when that DNA is disturbed or it is getting digested, okay, this process of transformation will not happen. And so they concluded that the transforming principle of Fredrick Griffith is DNA. And they told that DNA is the genetic material. So, in your question, in your board exam, if the question is explain the transforming principle, you have to explain Fredrick Griffith's experiment. Okay, but if the question is explain the experiment which proved the biochemical nature of transforming principle. So that word you should look for, children. If this question is prove the biochemical nature, then you have to think about the three scientists, Avery, McCarty and MacLeod. And this experiment you have to say. So this experiment you have to say in two parts. One is they have injected the molecules separately and then what happened. And the next part you have to say about this addition of enzymes one after the other and then what did they conclude. So they gave the biochemical basis of transforming principle and they also told that it is the DNA which is the genetic material. But some scientists, they did not accept. They told that okay, DNA is a transforming principle you have found out. But then we cannot accept that DNA is a genetic material with this experiment. But then the question was why they couldn't actually take from this experiment that DNA is a genetic material. They told what the scientists they gave the explanation is, if you want a molecule to be called as a genetic material, it should pass on the characters from parents to progeny. But here the character is not passed on from parent to progeny. It is passed on from one type of bacteria to another bacterium. Okay, so with this experiment we cannot prove that characters are passed on from parent to progeny or DNA is responsible for passing on the character from parent to progeny. Yes, it is right that it is giving that rough bacterium a smooth appearance. It is giving the information to produce the polysaccharide coat. That's true. But with this experiment, we cannot prove that DNA is a genetic material. And then there were two scientists, Hershy and Chase, who actually gave an unequivocal proof. Unequivocal means a proof which cannot be challenged by any scientist. So they, Hershy and Chase, they gave the unequivocal proof that DNA is genetic material. And for giving that proof, they did not take the streptococcus or the mice. They took some other experimental material. Now next, let's focus on that. So let's look into that unequivocal proof, the experiment done by Alfred Hershey and Martha Chase. They were the two scientists which in the year 1952 gave the unequivocal proof that DNA is genetic material. So the quest or the search for DNA material ended, or the quest for genetic material, which molecule is genetic material, ended with the experiments of Alfred Hershey and Martha Chase. So Hershey and Chase experiments. So as I told you, they did not experiment with the mice or the streptococcus pneumoniae. They took some other experimental organism. Now which one was that organism? They actually experimented on a bacterium, okay, and a bacteriophage. A bacteriophage. Now what is a bacteriophage? What is a bacterium? You all know. What is a bacteriophage? Yeah, they are the virus that infect a bacterium. That infects a bacterium. Or the virus whose host is a bacterium. Bacteria eating, or they go and attack a bacterial cell. So viruses which attacks the bacterial cell, you call such viruses as bacteriophages. So in short we can say Hershey and Chase were experimenting on virus and bacterium. Okay. So they actually studied the life cycle of virus onto this bacterium, or how this virus is affecting the bacterium. That they studied first. They studied that first. and then they framed their experiment. So let's also understand first, in short, how a bacteriophage is attacking a bacterial cell. Isn't that interesting to know? See, you have a bacterial cell. First, these viruses, they have just a protein coat. Viruses or bacteriophages, they have a protein coat. I am drawing the protein coat like this. Okay. And then they are having a genetic material. So these are the only parts, only two parts. One, they are having a protein coat. And the second one, they are having the DNA. This is the genetic material. This is the only two parts which are present in a bacteriophage. Okay. And these scientists, they wanted to find out how this bacteriophage is infecting a bacterial cell. So what happens is, this bacteriophage, first with this genetic material, it gets attached to this bacterial cell surface. It gets hold on to this bacterial cell surface. And then slowly it injects this particular genetic material into this bacterium. Genetic material is injected into this bacterial cell. And then what happens is, this genetic material of this bacteriophage or of that virus makes this bacterial cell to dance according to its tune. That means this particular bacterial cell, this bacteriophage which has inserted this genetic material, that genetic material goes and utilizes the machinery of the bacterium for its own means. Okay, so what happens is, this genetic material is there inside, it starts multiplying. It becomes plenty inside. The genetic material replicates. How? Using this bacterium, it's bacteria's machinery. Okay, and then after the genetic material is replicated, each of this genetic material will now also get a protein coat. It is getting a protein coat. Now you can see there are plenty of bacteriophages inside this particular bacterial cell. And then after some time, this bacterial cell, it will burst open. It will burst open. Okay. Or you say it will undergo lysis. Lysis means bursting open. Okay. To get devastated into pieces. Lysis means breaking down. So this is bacterial cell, it breaks down into pieces, and so all these phage particles are now free, it comes out. And then it goes and affects the neighboring bacterial cells. How? Again gets hold on to one particular bacterial cell, injects its genetic material, multiplies the genetic material, forms a protein coat, breaks open the bacterial cell, comes out. So this is the cycle, or this is the way a bacteriophage infects a bacterium. This life cycle of a bacteriophage inside a bacterium was actually studied by Hershey and Chase. And then they devised the experiment to find out how DNA is, how we can make this DNA to be called as genetic material. Because bacteriophage is having these two parts, DNA and proteins. They need not isolate many molecules like Avery McCarty and MacLeod had done. Okay, so for that they framed like this. So first they studied the nature of how bacteriophage is affecting a bacterium or infecting a bacterium. Now next they framed their experiment. So let us see how they framed their experiment. They took two sets of phage particles. Okay. In the first set, they radiolabeled the protein code. In the second set, they radiolabeled the DNA. Now I have used two terms, radiolabeling. What do you mean by labeling of a molecule? Suppose you have a notebook of yours, okay, and suppose teachers ask you to submit your notebooks. You all might have covered your notebooks with brown cover and you are submitting it. So then the class leader, after the teacher has corrected the book, the class leader is coming with the books, say 10 books, and leader is just distributing the books to you. How will the leader understand which is your book? Because there is a label kept with your name. Isn't it? Just reading the name, the leader can identify that it is your book and it will be handed over to you. So labeling is done to identify a molecule. Like it is done to identify whose book is that. So labeling in molecular biology is done to identify a molecule. So in order to identify a molecule, we have to find out the difference between molecules which we are going to identify. Here, Hershy and Chase wanted to identify or wanted to radiolabel the protein code with that of the genetic material. They wanted to identify the protein code with that of the genetic material. Because protein code and genetic material are only or DNA. Now I cannot call genetic material now, it is DNA. Protein code or the DNA are the only two parts. Okay, so they wanted to find out, or they wanted to differentiate between these two. They wanted to identify the movement of these two. What is happening to DNA? What is happening to protein code inside? They wanted to identify that. Okay, so in order to identify that, what they did is they radiolabeled the DNA. That means they had added a radioactive material to the DNA and they had added another radioactive material to protein. But there should be different materials, isn't it? Let's see. They radiolabeled their protein, I told you. Protein, as you know, it is made up of amino acids, isn't it? Proteins are made up of amino acids. So, amino acid, if I write like this, amino group, acid group, R group, H group. This R is variable. So, this is the general structure of amino acid. So, amino acid, they have carbon. Okay, carbon is there. Carbon is there, hydrogen is there, nitrogen is there, oxygen is there, carbon, hydrogen, nitrogen, oxygen, and some of the amino acids, they are rich in sulphur, so sulphur is also there. Okay, and methionine, cysteine, these are the amino acids which are rich in sulphur. All proteins will have this sulfur-rich amino acids inside them. So these are the components present in proteins. Okay. Now which are the components present in our DNA? DNA, as you know, it is made up of 3 components. Ribose sugar. So in that sugar, you are having carbon, hydrogen, oxygen. Isn't it? So ribose sugar is there. Then nitrogenous base. Second component of DNA is nitrogenous base. In that nitrogenous base, again carbon, hydrogen, oxygen, and apart from that, nitrogen is also there. The third component of DNA is phosphate. In that component, phosphorus is there, and then oxygen, and then hydrogen is there. So here I have listed down the various elements which are present in protein as well as in DNA. Now from this, if you want to differentiate or if you want to label protein from DNA, Okay, which radioactive material will you use? Will you use radioactive carbon? If you use a radioactive carbon here, then that radioactive carbon is present. Carbon is present with DNA as well as in proteins. So radioactivity will be shown in both DNA and proteins. That is, if you grow a bacteriophage in radioactive carbon, if you are growing a bacteriophage in a medium containing radioactive carbon, this carbon, since it is there in both protein and DNA, both the DNA as well as the protein coat will take up the radioactivity. So you cannot differentiate between the protein and the DNA. Protein coat and the DNA. Now next, if you are adding, instead of radioactive carbon, if you are adding radioactive hydrogen, in this case also, since hydrogen is present in both proteins as well as in DNA, both the protein coat as well as the DNA will take up the radioactive hydrogen from that medium in which it is growing. So both will have the radioactivity. Again, you cannot differentiate. Third component, nitrogen. This is also present in both proteins as well as DNA. So you cannot radiolabel with that also, because there is no point in doing. Both protein coat as well as DNA will have radioactivity. But then you cannot differentiate between them. Now next, if you take for radioactive oxygen, isotope, heavy isotope of oxygen, if you radiolabel with, again both the protein coat as well as this DNA is having. So, Hershey and Chase took the last option, sulphur and phosphorus. When they added radioactive sulphur, the sulphur is present only in the protein coat. It is absent in DNA. So, when the bacteriophages were grown in radioactive sulphur, only their protein coat got the radioactivity. Okay, so when they were grown in radioactive sulphur, only their protein coat was radiolabeled. It showed radioactivity. DNA did not show any radioactivity. Because sulphur is present only in protein coat, it is absent in DNA. Now in the next case, when another bacteriophage was there, and this bacteriophage is grown in a medium containing radioactive phosphorus. Now where is phosphorus present? Only in DNA it is present. Okay, and so the radioactivity was also present only with the DNA. So like this, they could produce bacteriophages, one having radioactive sulphur and the other one having radioactive phosphorus. So that is what is written here. Alfred Hershey and Martha Chase, they took a set of bacteriophage which was labeled with radiolabeled, its protein part was radiolabeled, and the other one, another set of bacteriophage whose DNA part was radiolabeled. I think from this explanation you understood why sulphur was taken here and why phosphorus was taken here, and that forms an important part of your board exam question also. Why Hershey and Chase took sulphur to radiolabel the protein? So the answer is sulphur is the only element which is present in protein and absent in DNA. And why then phosphorus to radiolabel DNA? Because phosphorus is the only element which is present in DNA and absent in protein. And radiolabeling is done to differentiate between DNA and protein. And so they were taken. Okay, now what is that radioactive material? Now I told you sulphur was radiolabeled and phosphorus was radiolabeled. Okay, so it is phosphorus 32, P32, and S36. These are the radioactive forms of sulphur and phosphorus. S36 and P32. Okay, so now let us come back to that experiment. So first they took a bacteriophage whose protein coat was radiolabelled. So radioactivity, radiolabelled part I am drawing with red color. So protein coat is radiolabelled here. Okay, DNA is normal. So which component was used to radiolabel this? It was radiolabelled with S36 isotope. S36. Now in the second case, protein was normal, that is it was grown in normal sulphur containing medium, but in this medium the phosphorus was made radioactive, and so the DNA got the radioactivity. It was radiolabelled with phosphorus 32. It was grown in P32 medium, so the DNA is radiolabelled here. Okay, now next what they did is they made these two phage particles to infect the bacterial cells. Okay. And the bacterial cells, the phage particles took hold over the bacterial cells and they injected the genetic material. The genetic material started multiplying inside and then the protein coats are formed. Okay, and then what happens? The whole bacterial cell is broken open, that it undergoes lysis. But Hershey and Chase, what they did is first they made these radiolabelled DNA and radiolabelled protein, these two phages to infect. So first phase is infection. After infection and before lysis, that means before the bacterial cell were actually broken and the new phage particle comes out. So before lysis, after infection, before lysis. This you should all keep it in mind when you write for the exam. Okay, experiments were done and after infection, before lysis, these particles were taken and they were blended. When did they do this? After infection. Okay, so after infection but before lysis. Before these bacterial cells were broken open, they were taken and they were blended. Okay, see, this is now sitting here inside. Like this, so it got infected. Okay, and then bacterial DNA, the phage DNA is inside here, and the phage DNA is inside, and here also the radiolabelled phage DNA is inside. Okay, they are inside here. They have infected, and then what happens? Next step is they form the protein coat here. So over each the genetic material there will be a protein coat. Here also protein coat is there. After infection, before lysis, this next step is it is blended. Means it is slowly shaken. Okay, slowly it is shaken. The whole arrangement is shaken. So here this DNA is now no more here. It has entered. Here also the DNA is no more here. It has entered. Okay, so after infection, before lysis, the whole arrangement is now blended. Slowly shaken. And then they are centrifuged. What is this centrifugation? they are subjected to very high speed rotation. Okay, revolutions per minute is controlled inside a centrifuge machine. They are centrifuged. And why they are centrifuged, children? Centrifugation is a method of separation based on the density. Okay, so which particle will be denser? Whether it will be the bacteriophage or the bacterium. Whether a virus will be denser or a bacterium will be denser. My question is, which one will be heavy? A bacteria or a virus? Naturally, it is the bacterium which is going to be heavier than the virus, isn't it? So when this is getting blended, what happens is this bacteria is getting separated from this virus particles. They are getting separated. Now they are separated after blending. So after they are getting separated, they are subjected to centrifugation. So naturally, which will come at the bottom? Because bacteria is heavy, bacteria will be there at the bottom. Okay, here also, in this case also, since bacteria is heavy, bacteria will be there at the bottom. And what about the virus? Virus will be there at the top. Here also, virus is there at the top. Okay, so they wanted to find out where is the bacterium and where is the virus. For that, they were looking for that radioactivity because they had radiolabelled it. Okay, so here they found out that the radioactivity is on the top. The radioactivity is in the top. Here is the radioactivity. So after centrifugation, you don't use the word top and bottom. Instead, after we do centrifugation, there is a word used. This is a vial. Vial means a small tube into which the centrifugation is done. Inside this tube, whatever has to be centrifuged is placed in a buffer solution and it is kept in a centrifugation machine opposite in the direction to equalize the volume, to equalize the weight, and they are rotated at very high speeds. After some time, the revolution is stopped, okay, and you take out the vial, that means these small tubes, and you can see something settled at the bottom and some at the top. So this is all based on their density that is getting settled. Simple, like when you take water and you put some sand and you stir it, sand will settle at the top, bottom, and some light particles will be there on the top. Same is here, but it is done using a machine. Because these are very small particles, it has to be rotated very fast. That is why. Okay, so this is a vial. After centrifugation is done, whatever material is there at the bottom, you call this material as the pellet. And whatever is there at the top, whichever is there at the top, you call this region as the supernatant. Supernatant. So after centrifugation, whatever is the material that you centrifuge, you will get a supernatant and a pellet. So here Hershy and Chase was finding out where is the radioactivity. So their question is, where is the radioactivity? Where is the radioactivity? This is a big question for them. Is it in the supernatant or is it in the pellet? So in this first case, they found that the radioactivity is there in the supernatant. And the second case, they found out that the radioactivity is there in the pellet. Pellet is showing radioactivity. Pellet is showing the radioactivity. Because this bacteria, it is having all the DNA molecules here with the protein coat. So, in the first case, they saw that supernatant is showing the radioactivity. Supernatant means it is a layer on the top, means the layer which is having light particles. So, among bacteria and the phage particles, which is lighter? The phage particle is lighter. So, they concluded that the phage particle is lighter and so phage will be there on the top. But which part of the phage is on the top? It is the protein coat which is there on the top. Because they had labeled the protein coat with sulphur. So they concluded that protein did not enter into this bacterial cell. If protein had entered into this bacterial cell, since bacteria is there on the pellet should have shown the radioactivity. Okay, if this protein coat has entered into this bacterium, so that the bacteria should show the radioactivity. So where will be the bacteria? Naturally it will be there at the pellet. Why? Because bacterial cells are heavier than the phage cells. So whatever molecule is there inside the bacterium, it should be there at the pellet. Okay, but then was any radioactivity in the pellet? No. Because then where is the radioactivity in the supernatant? Why? We had radiolabeled with protein, and what is the inference? Protein did not enter into the bacterial cell. But in the second case, the radioactivity is there inside the bacterial cell. The second case, which molecule was used to radiolabel? It was the DNA which was radiolabeled with phosphorus. So, since radioactivity is shown at the pellet, we can understand that radioactivity is there in the bacterial cell, and so the DNA which we have radiolabeled has entered into the bacterial cell. And that DNA which has entered into the bacterial cell is actually producing more and more phage particles. So the DNA is actually getting converted, or it is transferred from parents to progeny. So DNA is the genetic material. So but our biochemical basis, that is Avery McCarty and MacLeod, their conclusion that DNA's genetic material was not accepted because they did not have any parent-progeny relationship. But here you see this bacteria, this phage is entering into the bacterium and is producing many progenies of its own kind. Okay, and because of this, it was given that DNA is the final proof as DNA is a genetic material. So here in this experiment, you have to keep a few points in mind. First thing, the experimental material is totally different. It is the bacteriophage and the bacterium. Second thing, they had studied the life cycle. Third, they had radiolabeled the DNA with phosphorus P32 and sulphur and the protein with sulphur, that is S36. They radiolabeled them in order to track their path. Which one is entering into this bacterial cell? Okay, so in the first case they found that the radioactivity is there on the top. That means it has not entered into the bacterial cell. So that is not the genetic material. The second case they found that radioactivity is there in the pellet, or radioactivity is absent in the supernatant. And then they concluded that it is the DNA which is entering into the bacterial cell. And that is responsible for producing more and more phage particles. And so DNA is a genetic material, and that proof was unequivocally given. Okay, I hope you have enjoyed today's session. You had understood every part of it. And students, please understand that there are very minute questions which can be asked from this, like why sulphur and phosphorus was chosen. Okay, why there is no radioactivity here in the supernatant, and who gave the biochemical basis, who proposed the transforming principle, and how it was done. All those small things can be asked. Okay, so in the next session we'll come to another aspect of our DNA molecule, like how one DNA is becoming two. Like we know how one cell is becoming two. Similarly, how one DNA is becoming two, or the concept of DNA replication. And for that, keep subscribing the channel and keep watching the playlist. Go to the playlist. You can just play one by one the topics. Okay. If you haven't seen the previous classes, you go through that. Have a clear base, have a clear understanding of our genetic material, because now onwards we will be going on to how our DNA is working inside a cell. So keep asking your doubts, enrich yourself with the knowledge that is provided here. Keep asking doubts. Let us share knowledge through our channel. Keep watching, keep supporting. Thank you all.